The million-dollar assembly line. How generative AI actually invents new drugs. Most people think AI drug discovery is a single, massive neural network that magically spits out a cure for cancer. You type a disease into a search bar, hit enter, and a perfectly formed medicine pops out the other side. That is a complete myth. The companies actually winning the AI drug race aren't building a single Oracle model. They are building an automated factory. Think of it like a Tesla gigafactory, but for biology. One assembly line finds the problems worth solving. The next line designs the physical solutions. And the final line predicts whether those solutions will survive the brutal reality of human biology. Today, we are stripping away the marketing fluff and going under the hood of Insilico Medicine, one of the world's undisputed leaders in AI-native drug discovery. This company has managed to scale up a pipeline of over 30 AI-designed drug programs, landing multi-billion-dollar deals with pharma giants like Eli Lilly. We're going to break down exactly how their core platform, Pharma.AI, works step by step, from analyzing raw genetics to writing brand new chemical structures atom by atom, to predicting clinical trial success before a single human patient ever takes a pill. Part 1. The $3-billion-dollar bottleneck. To understand why this matters, you have to understand just how broken traditional drug discovery is. Right now, bringing a single new drug to market takes between 10 to 15 years and costs upwards of $2 to $3 billion. And here is the kicker. 90% of drugs that make it to human clinical trials end up failing. They either don't work or they are toxic. Imagine running a car company where 9 out of 10 cars you design explode on the test track after you've already spent a decade building them. That is the pharmaceutical industry. Traditional chemistry relies on trial and error. Scientists test tens of thousands of existing chemical compounds against a disease in a physical lab, hoping something sticks. It's slow, it's astronomically expensive, and it's mostly guesswork. Insilico's goal isn't to replace the laboratory entirely. It's to compress the first 5 years of this grueling process into a matter of months. They do this by breaking the problem down into 3 distinct AI systems: PandaOmics, Chemistry 42, and InClinico. Let's look at how the first machine in the factory operates. Part 2. PandaOmics, finding the disease's weak spot. Every disease has a master switch, usually a specific protein in the body that is malfunctioning. In biology, we call this a therapeutic target. If you can design a molecule to block or activate that protein, you can cure or treat the disease. Here's the catch. Human cells express around 20,000 different genes, translating into hundreds of thousands of protein variations. When a person gets sick, thousands of these genes change simultaneously. If you look at a cancer cell, it looks like a chaotic crime scene. Traditional biology spends years trying to figure out which gene actually caused the crime, and which ones are just innocent bystanders reacting to the chaos. If you pick the wrong target, you will waste hundreds of millions of dollars developing a drug for a protein that doesn't actually drive the disease. This is where PandaOmics steps in. Its job is to find the disease's ultimate weak spot in weeks instead of years. PandaOmics doesn't just look at one type of data. It consumes millions of massive, messy biological data sets. We are talking about RNA sequencing, DNA sequencing, proteomics, metabolomics, and massive clinical trial databases. But it also does something highly unusual. It reads the entire sum of human medical literature. It ingests millions of scientific papers, patents, and electronic biomedical databases. Under the hood, PandaOmics processes this mountain of data using 2 primary AI architectures. First, it uses graph neural networks. Imagine every protein in the human body connected like a giant trillion-node social network. Protein A influences protein B, which activates protein C, which suppresses protein D. Normal neural networks struggle with this kind of data because they look at data points independently. Graph neural networks, however, learn biology as an interconnected web. They look at the structural pathways of a disease, mapping out how a signal cascades through a cell. Second, it uses transformer language models. Just like large language models predict the next word in a sentence, these biological transformers predict cellular relationships. By reading millions of research papers, the AI learns text-based associations that no human could map out in a lifetime. It connects a hidden mention of a protein in a 2012 oncology paper to a genomic dataset published in 2025, realizing they are talking about the exact same pathway. All of this data is funneled into a massive knowledge graph. The AI reasons across this graph, linking a disease to a gene, to a protein, to a historical drug, to a clinical outcome. Instead of forcing scientists to guess which of the 20,000 genes to target, PandaOmics spits out a ranked list of the top 3 or 4 highest probability therapeutic targets. It narrows the entire biological search space down to a single, mathematically validated bullseye. OneEarth Part 3, Chemistry 42, the AI Molecular Architect. Once PandaOmics hands over the target protein, the second machine in the factory turns on. This is Chemistry 42, and its job is to build the actual drug. In traditional medicinal chemistry, humans look at libraries of existing molecules. They might synthesize 50,000 slight variations of a known compound in a physical lab, testing them one by one to see if any of them bind to the target protein. Chemistry 42 flips the script. Instead of searching through what already exists, it uses generative AI to invent brand new molecules from scratch. Think of it this way. If a regular language model predicts the next word, Chemistry 42 predicts the next atom. It draws entirely new chemical structures that have never existed on Earth before, specifically tailored to fit into the target protein like a key into a lock. To do this, the AI has to speak the language of chemistry. It processes molecules in multiple formats simultaneously. It looks at them as text strings. It looks at them as 2D graphs where atoms are nodes and chemical bonds are edges, and it analyzes them as highly complex 3D physical structures. But you can't just use one AI model to design a drug, because drugs are incredibly complicated. Chemistry 42 actually utilizes an ensemble of dozens of different generative models working together, including generative adversarial networks, variational autoencoders, diffusion models, and transformers. Each model explores chemical space in a slightly different way. The core engine driving this creative process is reinforcement learning. Imagine an AI playing a video game, but instead of trying to get a high score in chess or Super Mario, the AI gets points for designing a perfect drug. The system sets up strict reward functions. It awards points if a generated molecule binds tightly to the target protein, but it subtracts points if the molecule is toxic, if it's too difficult for a human chemist to synthesize in a lab, or if it resembles someone else's patent. Every time the AI generates a batch of virtual molecules, they are scored. The weak molecules are deleted. The high-scoring molecules are selected, mutated, and used as the parents for the next generation of designs. The AI repeats this loop millions of times, evolving the drug candidate in a matter of hours. What makes this incredibly difficult is what scientists call multi-objective optimization. In drug discovery, your goals are constantly fighting each other. A molecule that binds incredibly well to a disease might also be highly toxic to the liver. A molecule that is completely safe might be instantly destroyed by the stomach before it ever reaches the bloodstream. Chemistry 42 acts as a master negotiator, balancing dozens of conflicting variables, from binding affinity and toxicity to brain penetration and manufacturing costs, to find the ultimate chemical compromise. Before a single physical drop of liquid is touched in a lab, Chemistry 42 runs massive physics simulations. It models protein folding, calculates molecular dynamics, and tests virtual docking. It simulates how the generated molecule moves, bends, and locks into the disease protein. By killing off thousands of weak candidates in a virtual simulation, Insilico saves years of useless manual lab work. Part 4. InClinico, predicting the future of clinical trials. Now, the AI has found the target, and it has designed a pristine, novel molecule to attack it. Normally, this is where the AI's job ends, and the multi-million-dollar human clinical trials begin. But remember, 90% of drugs fail in human trials. This is the most expensive graveyard in science. Companies spend hundreds of millions of dollars running Phase 1, Phase 2, and Phase 3 trials, only to realize years later that the drug doesn't work well enough in real human populations or has unexpected side effects. This brings us to the third piece of the factory, InClinico. InClinico is a predictive AI system designed to look into the future and estimate the probability of a drug's success before a trial ever begins. It trains on an immense archive of historical clinical trial data. It looks at decades of FDA outcomes, specific protocol designs, trial endpoints, enrollment statistics, patient demographics, and genetic biomarkers. When a company is preparing to launch a clinical trial, InClinico evaluates the entire setup. It doesn't just look at the molecule, it analyzes the sample size of the trial, the specific severity of the disease in the chosen patient population, whether there are clear biomarkers to track if the drug is working, and even the historical track record of the company sponsoring the trial. Using advanced machine learning models, like gradient boosting, deep neural networks, and Bayesian inference, InClinico processes hundreds of these variables to output a calibrated probability score. For example, it might look at a protocol and state that this specific trial has a 67% chance of passing Phase 2. Instead of relying on executive intuition or gut feelings to greenlight a multi-million-dollar trial, pharmaceutical companies can use quantitative risk estimates to prioritize only the absolute strongest drug candidates. Part 5. The Data Flywheel and the 2026 Reality. When you connect these three systems, you get a continuous pipeline. Patient data goes into PandaOmics to find a disease protein. That protein is fed into Chemistry 42 to design a candidate drug. That drug is validated in a physical lab, and the entire package is evaluated by InClinico to optimize the human trials. This integrated assembly line is exactly why the world's biggest pharmaceutical companies are paying billions of dollars to partner with Insilico. Eli Lilly expanded its massive collaboration in a deal worth up to $2.75 billion. Other major players, like Takeda and SK Biopharmaceuticals, have locked in massive multi-billion-dollar partnerships to plug Insilico's AI engine directly into their own pipelines. They aren't buying a single drug, they are buying into a data flywheel. Every time an experiment succeeds or fails in the physical lab, that data is fed right back into the AI models. The system gets smarter with every single molecule it designs, but let's wrap this up with a crucial reality check. AI cannot replace physical biology, a neural network cannot prove a drug is safe, and it cannot replace human clinical trials. Every single molecule generated by Chemistry 42 still has to be physically synthesized, tested on living cells, evaluated in animal models, and rigorously tested in phases of human trials before getting regulatory approval. The ultimate bottleneck in medicine isn't generating candidate molecules anymore. It's proving safety and efficacy in living human beings. What AI has done is completely shatter the early-stage discovery bottleneck. Insilico proved this by taking their lead drug candidate for idiopathic pulmonary fibrosis from an absolute blank slate to mid-stage human clinical trials in record time for a fraction of the traditional cost. We have officially entered the era of AI-native biotech. The drugs being tested in hospitals today are no longer just the product of human trial and error. They are the products of an AI factory floor.